welcome to this plenary session. I have a huge honor in being able to introduce Professor Pedro Jordano of the Spanish Council of Scientific Research at the Estación Biológica de Doñana. Pedro is a giant in the field, whether you consider the field to be ecology, conservation, Spanish science, or world science. I could go on at length about his many honors, but instead I thought what I would do is just tell you why I am so excited every time I open a journal issue and see one of his papers. Because I know I'm going to get one of three things, and probably all of them in combination. One will be really exciting data about the most exciting questions that ecology and conservation ask. In particular, his career-long studies of interactions between plants and the animals that disperse them have fundamentally changed our understanding of how mutualisms work. So that's the first thing. The second thing is his incredible work in the conceptual areas of ecology, particularly community ecology. And here I have to mention the incredible innovations of ecological networks theory, which he is almost single-handedly responsible for starting and continuing at a worldwide scale. And this has fundamentally changed our understanding of ecological systems, communities, and species interactions in a remarkable way. So data conceptualization. And then the third is integration. He's remarkable at integrating different perspectives and different ideas to give new insights. So for example, his perspectives on species interactions have always been integrative between plants and animals. And I think you'll see that in his talk today. But more than that, he's an integrator of people. And I think this is reflected in his interest in ecological networks. He brings people together, he brings teams together. This is very evident in the study of ecological networks, where Spanish scientists and Latin American scientists in particular are absolutely the lead in this, and this is very much due to his influence. I think you're going to see all of these traits of Pedro's come together today in his talk. His fascination and eye for data, his ability to conceptualize a problem, and his integrative sense, both between ideas and among people. And all of this together have allowed him to become a passionate voice for conservation. So his talk today is entitled Cryptic Disruptions of Plant-Animal Mutualisms in the Anthropocene, and I give you Pedro Jordano.
Thank you. Okay, good morning everyone, and thank you very much, Judy, for the nice words and the generous introduction. And I'd like to thank also, express my sincere appreciation to the organizing committee for inviting me to be here today. It's really an honor, it's a privilege, and as John said before in his keynote, I'll never forget this. It's really very impressive, and I didn't expect that invitation, so I was very pleased to come, and certainly I'm very happy to be here and have this opportunity to share my ideas with all you. So thank you for coming and for your attention. I very much hope that you will have a fruitful Congress and also a very nice stay here in Spain. So today what I want to do is to review a particular form of biodiversity, and this is the biodiversity of ecological interactions. And so my idea is to give a very brief introduction to the web of life, the whole set of ecological interactions that support biodiversity, that support species richness. There is no single species on Earth that lives without interacting with other species. So we need ecological interactions. We need to understand ecological interactions among plants and animals to understand plant population dynamics, plant expansion ranges, plant colonization abilities, potential for response to climate change, and a series of other important aspects. So my first short part of the talk will be dedicated to describing these biodiversities, what I call the biodiversities interactome, the whole set of biotic interactions that we can catalog, we can record and see in nature, on Earth, among species on Earth. And then in the second part of the talk, I'll overview a series of case studies illustrating what are the consequences of this extinction of this biodiversity. What are the consequences of extinction of the biodiversity of interactions? So myriads, literally myriads of different ecological interactions support plant life. In plant communities, many plants depend strictly on interactions with animal species and fungi for their survival. So what we have in local assemblages of species is a very quick increase in the richness of interactions when we increase species diversity. So when we increase species biodiversity, species richness, we are increasing also the number of interactions. And we may find hyper-diverse communities where even more than 1,000 type of species, plants and pollinators, for instance, can be found in temperate forests in Japan, and these up to literally thousands of different ecological interactions. So this region of interactions need to be recorded. We need to be able to find ways to formalize descriptions, useful descriptions of these components of biodiversity. So let me turn to case studies that we have been studying in the Mediterranean. And here in the Mediterranean ecosystem, as John has been brilliantly exposed in his previous keynote, we have many different, a very high diversity of frugivorous animals. So here you have frugivorous birds that they vary a lot in the degree of dependence and reliance on fleshy fruited species. And we have in the Iberian Peninsula around 120 fleshy fruited species that is producing fruits that are specifically adapted to endozoocorous seed dispersal by animal frugivores. And among the frugivores, we not only have frugivorous birds, also mammals, different species of mammals, reptiles, and even fish species. So all those species are the partners that support the natural regeneration of the populations of those plants in Mediterranean habitats. We have, for instance, the mesocarnivores here, genets, badgers, stone martens, red foxes, that at some period of the year, during their annual cycles, they can rely quite heavily on fruits, and they perform a very important role in the seed dispersal process of plant populations in the Mediterranean. So really cataloging and prospecting and sampling this biodiversity of interactions is extremely important. We need to understand how this biodiversity is structured. For that, we are using different methods. For instance, here, I'm just summarizing different photographs of taken with camera traps that passively monitor visitation by avian and mammal frugivores to the plants. We are also using, of course, direct watches in the plants to record interactions, and also genetic techniques that I'll turn later in the talk to describe more in detail. So all those methods go along the line of sampling biodiversity, but our sampling of biodiversity, our specimens that we are sampling are pairwise interactions among animals and plants. We are no longer interested in specific species, we are interested in the pairwise interactions among species. So we go out to the field to grasp this biodiversity. And that poses exactly an analogous problems and limitations to the sampling of species diversity. So all this richness of ecological interactions results in the building up of highly complex networks of interactions. And here we have just a couple of representation, like snapshots of interactions between fleshy fruited plants are the nodes in green in this complex network, and then different animal species, frugivorous animals that consume the fruits and disperse the seed in a lowland site in Doñana, in a highland site in the mountains of southeastern Spain. So in the last 25 years, we have been exploring together with Jens Olesen and Jordi Vasconte, bringing tools from mathematical topology and from structural statistics in physics to analyze the complexity of these networks, to describe them, to analyze statistically this complexity, and then to develop forecasting models to answer the question of what are the consequences of losing specific interactions or specific species in those networks. So when we zoom in those thin links that unite two of those nodes, a plant node and an animal node, what we have is a mutual relationship of interdependence. So each interaction involves interaction of dependence of the plant on the animal for successful dispersal of the seeds, and then also a dependence of the animal on the supply of food that the plant provides, and the energetic and nutritional supply of food. So those are the details that are behind those interactions, and they hide an extremely rich of natural history details. Today, I'm going to zoom in those details to explore the consequences that we have when losing those ecological interactions. So let me show to what I call the anatomy of an interaction. Among free-living species, like fleshy fruited plants that live outside in nature, and free-living animal frugivores, the interactions occur by means of interspecific encounters. So interactions that we can sample in nature are encounters between individuals of plant species and animal species. And they have a probability of interspecific encounter that determines the frequency of occurrence of interactions that we can record in natural habitats. But also they have an immediate outcome that usually is what we see when we observe plants being pollinated by insects or frugivorous animals dispersing seeds in the field. And here we can see the immediate outcomes in terms of successful flower fertilization or successful dispersal of the seeds or seed predation or fruit damage and dropout beneath the parent plant. But then interactions, those ecological interactions have also delayed outcomes. So to speak, they have consequences that go beyond after the interactions actually occurred in nature. So we urgently need ways to adequately format and record and archive the data on interaction biodiversity. In the last year, we have been working with the biodiversity information standards within the task group on biological interactions data to try to develop what we call ontologies for the recording of interactions in databases like GBIF and other bases. So our goal is to, in collaboration with the Brazilian network of plant pollinator interactions, we are developing a vocabulary, an ontology, to try to describe every element that we can record in nature about an ecological interactions, about the consequences of the interactions and the frequency of occurrences. And this is difficult. This is a difficult task, taking into account the enormous diversity of interactions and the richness of natural history details that they have. So we developed different descriptors for the taxonomy, the IDs, and information on functional traits about the partner species, but also descriptions of the encounter rates, could be binary, could be quantitative, and descriptors whenever information is available about the immediate outcomes of the interactions and the delayed outcomes of the interactions. So we are urgently needed this kind of information. Biodiversity is not only a species, it is also the framework that supports the lives of a species, that supports the lives of all those species, and those are the ecological interactions. And that is urgent because what we are facing with biodiversity loss is a more insidious type of extinction, like Professor Daniel Jansen said, is the extinction of ecological interactions, not just the extinction of a species, but the extinction of interactions among them that may precede the extinction of the actual species. Interactions become extinct when they are no longer functional. When a species might be present, we can get the species present in our inventory in a fragment of tropical forest. We may see the tracks of the tapir, or we may hear howler monkeys. We can record a toucan flying, but the population densities of those animals that have this ecological service are so low that they are no longer functional. The species are locally extinct, the interactions are gone. And the type of interactions and functional services I'm talking about are mostly related, in the case of plants, to the movement of gametes and the movement of propagules through interactions with animal vector for pollination and for seed dispersal. So those are very important components of those interactions. What happens when those interactions are lost? Let me review and discuss with you several studies that we have done in southern Spain, Canary Islands, and the Atlantic rainforest in southeastern Brazil, to try to understand what are the consequences of forest fragmentations, of defaunation, when those interactions are extinct, those interactions become extinct. Let's move to the Canary Islands in Spain, offshore of the coast of Morocco, and we'll concentrate here in the three central islands, and together with Nestor Pérez Méndez, Cristina Garcia, and Alfredo Valido, we have been studying the seed dispersal of a narrow endemic of these three central islands in the Canaries, Neocamelea pulverulenta in the family Neoraceae. Neocamelea pulverulenta is present in the three central islands, in the Canary Islands, and it is a plant species that produces those relatively large drupes, larger than 14 millimeters in diameter, with relatively large seeds, and it is exclusively dispersed by the giant Galotia lizards in the Canary Islands. They are the exclusive disperser of this plant. No other frugivore in the island dispersed Neocamelea seed, probably because of their relatively large size that makes it difficult for small-bodied frugivores to handle and to ingest the fruits adequately for an efficient seed dispersal. So giant lizards are the sole disperser of the plant. But then the lizards, they have a strongly asymmetric interactions with the Neocamelea, because we have records of the lizard dispersing up to 50 fleshy fruited species in the Canary Islands. So they are super generalist frugivores. So, sorry, I forgot to mention that the plant is self-compatible, has an extremely interesting breeding system, and the main pollinators are a small group of species of bibionid flies and califorous flies, a couple of alictid bee species, and anthophoric bee, and 2 ant species that visit the flower and pollinate the flowers. So the genus Galotia is endemic to the Canary Islands, and they have a very nice radiation on the islands. So starting with the now extinct Galotia goliath, that was extinct around 500 years ago. It was a giant lizard the size of an iguana, and was extinct after the arrival of humans. The first arrival of Aboriginal Guanche people in Canary Island took place around 2,500 years ago, And then with the arrival of the Europeans 500 years ago, they decimated many populations of those giant lizards. So we have lost this Galotia goliath, but there are now four giant lizard species in the Canary Islands, yet 3 of them are at the edge of the extinction. In fact, 2 species, Intermedia and Gomerana, were, we believe they were extinct and were quite recently rediscovered in very small, highly isolated, very few individuals populations, no longer functional for seed dispersal. Then we have the medium-sized Galotia galoti, which is strongly frugivore and able to disperse the seed of Neocamelea, and then 2 additional small insectivorous species, Cesaris and Atlantica, that don't consume fruits. So nowadays, the main frugivore dispersing efficiently the seeds of Neocamelea is Galotia stellini, and we have among the 3 islands a very interesting defaunation gradient coming from the Gran Canary Island, where the populations are not defaunated and the plant is still dispersed by a Galotia stellini, a form of stellini that is quite similar to the subfossil stellini for which we have records. Then, intermediate in the defaunation gradient in Tenerife, the plant is still efficiently dispersed by Galotia galoti, the medium-sized lizards. And then in the defaunated islands of La Gomera, Galotia cesaris is unable to handle the fruits, and the dispersal of Neocamelea, we can be considered, is completely collapsed. As we will see in a minute. So when we analyze plant frugivore interaction data, we are based not only on observation of the interaction, but also assessing the consequences. A technique that we are using quite heavily in our studies of seed dispersal in Mediterranean areas is using genetic tools to obtain DNA amplification of two types of DNA. We are obtaining DNA of animal origin that is just swabbing the surface of dispersed seeds that we can sample, either defecated or regurgitated by the animals, and that we can sample in the field with seed traps that we collect to passively monitor seed grain. We extract this material to amplify sequences of DNA from animal origin, and then using DNA barcoding techniques to identify the frugivore species dispersing that particular seed that we sample in a particular part of the habitat. And we are doing that with other species dispersed by highly diversified assemblages of avian frugivores. Here we have a single species, so we are not using this technique. We are using the other DNA extraction that we do. We extract and amplify DNA from the woody endocarp of the seed. The endocarp of the seed is a diploid tissue that derives from the carpellar walls of the flower, and it's a multilocus genotype. The multilocus microsatellite genotype of that tissue are identical to the multilocus genotype that we can sample in the fruiting plant population by extracting DNA from the adult plants. So typically we have in each island plots of 1 hectare where we have multilocus genotypes derived with hypervariable DNA microsatellites for all the fruiting plants that are in 1 hectare of those plots, and they are hundreds of plants. So we build libraries of those multilocus genotypes, and then when we find a scat of a lizard with a seed, we amplify the DNA from the endocarp and compare to match to which individual plant that Galotia lizard was feeding. So we can determine the seed dispersal distances away from the maternal plant. So here, all the red bars here are different seed dispersal events performed by the lizards, and we can see here in this dispersal kernel is just a mathematical function showing up the probability of seeds dispersed away from source plant located here at 0, reaching down up to a given distance from the maternal population. So the dispersal kernel are a description of where are the seeds going from a particular maternal plant. And then we see that in the non-defaunated island, where seeds are yet functionally dispersed by Gallotia stellini, we still record long-distance dispersal events within the population. Of course, we genotype also seeds that we can match with any plant in our 1 hectare plot, and those could be immigrants from other populations. I intern later on this in a minute. So when we go to Tenerife, what we see is a truncation of the dispersal kernel. We have lost the long-distance dispersal events, and then most seeds reach shorter distances when seeds are dispersed by the medium-sized lizards. And in the totally defaunated La Gomera, Gallotia cesaris is not able to disperse efficiently the seeds, and then we see the dispersal kernel truncated to distances up to 3 or 4 meters from the parent plants, probably not by the actions of Gallotti, but by the action of runoff. So truncation of dispersal kernels occurs because of defaunation of the large frugivores, and that is probably a very common, a very frequent situation in many oceanic islands where downsizing of the frugivore assemblages has been extreme. We have lost the very large seed dispersers that are able to move the seeds away from the parent plants at very long distances. So that defaunation event, that loss of the large frugivores, also has signals on the overall genetic structure of the plant population. So here we have the population genetic networks, population genetic graph that depict the genetic distances, the conditional genetic distances, the conditional genetic dissimilarity among every pair of populations. So we have sampled up to 74 populations of Neocamellia in the 3 islands, and we built those networks, and it is pretty evident that in the defaunated island, the overall population graph is way larger than the one that we observe in the non-defaunated Tenerife and non-defaunated Gran Canaria. So even despite the smaller size of the La Gomera island, we see a much diversified, a greater difference, a greater level of population, differentiation among populations. So when we are losing the large lizard, probably we are losing also the probabilities of those very long dispersal events in the islands that occur because of the actions of predators on the lizards, as was brilliantly presented by David Padilla, Manuel Nogales, and Aaron Gonzalez years ago. So kestrel, common kestrel, they are able to feed and predate on frugivorous lizards, and those lizards have Neocamellia seeds inside them. We estimate that up to 85% of the lizard prey that common kestrels are capturing contains seeds in their interior, in the stomachs and intestine. When this male kestrel is taking the lizard to the nest, and we have located seeds and seedlings emerging of Neocamellia beneath the nest at the perching position of the kestrel. We have documented seed dispersal events up to 1.7 kilometers away from source plant due to secondary dispersal by these animals. Obviously, when the lizards disappear and the lizards no longer are consuming fruits and seeds, obviously the secondary dispersal of those seeds cannot occur, and then the truncation of the dispersal is evident. So moving to the southeastern coast of Brazil, to the totally different system, we can also evaluate what are the main drivers of this loss of interactions. And the main drivers of loss of interactions here in many areas nowadays, as the IPBES report pointed out, are deforestation and overhunting. And deforestation and overhunting are running in parallel, as we are going to see, and create the situations ideal for the extinction, the local extinction of functional interactions between plants and animals. So here we see the original station of the Atlantic forest in the state of Sao Paulo. This is the map of the state of Sao Paulo 500 years ago, and then the extent of the Atlantic rainforest have been decreasing due to deforestation, and now the remnants are preserved in the eastern ranges, in the sierras of the along the coast, and Serra do Mar and other areas. So here you have in the extent of the whole biome of the Atlantic rainforest in orange. So now what we have in green is 12% remnant Atlantic forest. Up to 80% of that remnant Atlantic forest is present in fragments of less than 50 hectares. So it's a very, very dramatic event of forest deforestation. At the same time, to fragmentation, it happens that hunting increases in pressure. So here you have, for a family of hunters in Serra do Mar, what are the frequencies of their prey taken during 1 year. 72 large crayfish or guans, 180 tinamous, and more than 400 toucans. A single family of hunters in a place in Serra do Mar. So overhunting pressure can create local conditions for defaunation and the disappearance of frugivores, so that we transition from those hyperdiverse assemblages of avian frugivores and vertebrate frugivores that are dispersing efficiently the plants to situations in the fragmented forest where only a small body size species like the thrush species remain. Those are quite small and then represent also a downsizing, as we have just seen with the Galotia lizards in the Canary Islands, but we are seeing here to a major scale, a community scale, where the downsizing can be quite important. For instance, we report a downsizing of 2.5 times in the gape width of the frugivorous birds, and that means that without decreasing in gape width, the frugivores that are remnant in the fragmented, in the defaunated forest, they are not able to efficiently disperse the large fruit and the large seeds that many hardwoods produce. We are going to see the consequences in a minute, and this is 7 times a reduction in size. Is that it's a similar type of reduction of downsizing that if you have a Rottweiler and you change that with a small pug, that would be a 7 times downsizing of your pet. Is a 7 times downsizing of the functional frugivores that these plants have. So what we find, together with Mauro Galetti and many other people collaborating in this project in southeastern Brazil, we have been sampling seeds and studying the seed dispersal of Euterpe edulis in the family Arecaceae. Is the Palmito Jussara, is an endemic species in the Atlantic rainforest. And what we find in relation to seed size, we see here the distribution, the distribution of seed sizes, and we see here each column represents different non-defaunated areas where the palm is still dispersed by large frugivores, like toucans, cotingids, and crassids, and in blue, and the seeds are significantly larger than the seeds that we can find consistently in fragments. You go to a fragment, you are not going to find large-seeded Euterpe individuals. You are going to find small seeds of Euterpe. And if you find large fruit, those will be pecked, will be below the palms, and will be quickly attacked by rodents and by fungi. So this is very consistent, and it is probably due to the strong selective pressures that defaunation has implied for the functioning of those euterpe plant populations. So in upland populations where the frugivores are extirpated, we are losing the large frugivorous animals that are able to swallow the large fruits and disperse the large seeds. So here in the pristine areas, this is a cubic spline illustrating the seed selection pattern by the frugivores in the pristine areas, and you can see that the whole range of phenotypic variation in seed size is adequately dispersed. Then when we turn to the selection function here, the cubic spline in orange for the defaunated areas, the probability of seeds being dispersed, if we compare the seeds that we found within fruits that are not dispersed with the seed size that we found in excrement of the animals or regurgitated by the animal frugivores, we see that the probability very quickly drops down to 12 millimeters, sorry, I said that in Spanish, 12 millimeters in size. And that is a magic size that happens to be the average gape width of the small frugivores that are remnants in the area. So a strong selection operating on seed size as a consequence of losing the large frugivore in the defaunated areas. We have developed, in collaboration with Matias Pires and other people, models of response to selection on seed size based on the narrow sense heritability of seed size determined by the multilocus genotypes of the plants, and then the selection differentials estimated by comparing the seed size dispersed in pristine areas and in defaunated areas. And we see that, surprisingly, very quickly, the response to selection can be very quick. And very quickly, we can reach the seed sizes, starting from the seed sizes that we find in pristine areas of the Ombrophilus Mata-Atlantica forest here in the upper part, and the semi-deciduous Atlantic rainforest to control for climatic differences within the biome. We may find a very rapid response in seed size decrease that may happen even within 100 years. So very rapid changes can be possible just because of this very strong response to the tremendous selection differential that is found in those populations when the major dispersal agents are lost. So from a pristine area where all the phenotypic range of seed size is dispersed, we go in the defaunated forest to only interactions occurring only with small frugivores that efficiently disperse just the small seeds, quite similar to the defaunation that we saw before. And that happened very recently in the evolutionary history of Euterpe edulis populations in southeastern Brazil, have been associated to the development and the intensive development of coffee plantations and sugarcane plantations, and also deforestation for livestock and grazing, and in the last 200 or 250 years. So those defaunation events that run in parallel also to the loss of forest have occurred very quickly. We used coalescent models to try to infer the time to the most recent common ancestor of the Euterpe populations that we are sampling here, to test whether or not the population differences that in seed size that we have seen between defaunated areas and non-defaunated areas could be attributable simply to different evolutionary history between those types of populations. In our generalized linear model, I forgot to mention that we included, in addition to the effect of the type of defaunation found in an ocar area, we also included a lot of information of variables on nutrient content in the soil, climatic data like rainfall, wind intensity, even data on the density of post-dispersal seed predators that could be influenced seed size. No one of these factors explained successfully the differences in seed size between the two types of populations, just the defaunation event. So with using the equiescent, we have estimated that the time to the most recent common ancestor preceded very much the most recent differentiation between the fragments. So the most likely explanation for the size differences that we see between the non-defaunated and the defaunated site could be attributed to the recent history of forest fragmentation and defaunation in those areas. So far, we have seen consequences. Wow. So far, we have seen consequences for the life history of single species, but obviously defaunation also brings consequences at the community. So we are starting with an initial community in a pristine area where we have all the whole flora present in our forest inventory. We have hardwoods that are highlighted here by dark blue, and we have also a whole representation of the whole diversity of the frugivore assembly. As we advance to the defaunated and fragmented areas, we are losing not only the large frugivores, we are losing also the large seeded and large fruited hardwoods. And that has consequences for many ecological and many ecosystem functions that occur in those rainforests. So we know that seed diameter, seed size, correlates positively with wood density of the adult plant obtained later on after the whole regeneration cycle is completed. So when we see in the forest fragment extirpated those large frugivores, they are invading the seed dispersal efficiency of the large seeded tree species. The hardwoods almost disappear. The Atlantic forest could harbor, if I remember well, up to 2,500 tree species. Up to 18% of those species have fruit size and seed size above 12 millimeters. That is this magic size that poses a threshold for the successful dispersal of the plants. So when those trees disappear, crucial ecosystem functions like the capacity for carbon storage disappear. We have tested that with the forest inventory data for 23 locations of the Atlantic forest, and we see here the trajectories simulated when we simulate increased percentages of fruit species of larger than 12 millimeters disappearing. So we are increasingly killing species of large seeded trees that happen to be the densest wood and also the larger capacity for carbon storage. So we see both trajectories point to a very significant, highly significant losses of the carbon stock potential of those forests when we move to defaunated areas where the hardwoods no longer recruit. Only softwood species with small fruits, small seeds, and not very dense wood occur. So this is probably happening in many different types of rainforests, and is one of the kind of transformation that we can see deriving directly from human-caused disturbances like forest loss and defaunation. Well, so far I have been talking just about seed dispersal interactions, but now we are considering how the multiple types of different interactions, both with mutualists and antagonists, they add up during the demographic cycle of any plant. So we know, or you know very well, much better than me, that any demographic cycle of a higher plant, as Professor John Harper brilliantly pointed in his fascinating book, Population Biology of Plants, is made up of different demographic stages, successional stages, transitions that imply interactions with different biotic partners. We need to understand the whole richness of those biotic influences, if we aim to understand what are the factors that limit that those probabilities of transition from one demographic stage to the other don't reach zero. And so I've been working quite heavily some time ago with St. Lucie cherries, 3 species distributed throughout Europe, but in fragmented populations in southern Spain. And here, I was just monitoring and recording all the type of interactions with herbivores and fungi. Here, with different types of pollinators, both bees and flies, and with animal frugivores dispersing the seeds. This is just a small example to illustrate the type of approach that we are using now to understand the whole set of biotic interactions. Any type of biotic interaction, what are the consequences for the plants, and then so that we can adequately forecast what would be the consequences for losing one of those interactions. We are doing that by using a complex network analysis technique that is called multi-layer networks, and these would be the appearance of the multi-layer network for the interactions of individual trees of those Prunus mahaleb populations with their biotic partners, the 3 types of biotic partners. So here we have a first layer depicting the pairwise interactions between individual trees that are the yellow triangles with all the coterie of antagonistic herbivores and fungi. Then we use a second layer illustrating the interactions of those same trees with the assemblage of pollinators, of insect pollinators. And then the third layer illustrates the interactions with the mutualistic animal frugivores for this population of Prunus mahaleb trees. And this type of multi-layer networks is called a multiplex network because a set of the nodes of the network that are the individual trees, they are repeated in each layer. So that we use those interlayer links among the individual trees to estimate what are the influences of each type of interactions on the definite effects that remains to the other interaction that comes later. So from herbivory on emerging leaves and buds and flower buds and flowers to the fruit set by the pollinators to the number of seeds finally dispersed with the interaction by the frugivorous animals. Multi-layer networks, let very briefly explain, use a type of mathematical structure to describe them. Those are called tensor. So a tensor of order 0 is just a scalar that represents just a pairwise interaction between an individual tree and a particular, let's say, pollinator species. A scalar and is that tensor of order 0. We have 10 tensors of order 1 are very familiar to all of us. They are vectors, and they represent what is the range, the full range of partners that any individual tree has. Is how, so to speak, how each individual tree, so to speak, builds its interactions with the whole biotic community. That is a vector. So if we have those vectors determined for all the trees in the population, we can stack them and we obtain the tensor of order 2, that is the famous adjacency matrices describing the interactions between the trees and all the biotic vectors. And then the higher order tensors of order 3, 4, or even higher describe just the structure that we have seen in the previous slide, the multi-layer network. So when we analyze that, we can represent the interactions between individual trees that are here, the rows in the matrix, with the interactions with all the biotic partners that are the different species of pollinators, frugivores, fungi, herbivores, etc. And it happens that we find groups of trees that have similar patterns of interactions with the whole set of biotic partners that are growing in specific locations. So the whole set of interactions of individual trees inhabiting areas of pine forest are quite different to the suite of biotic interactions that we find in trees that are isolated, growing isolated in the open grassland, or trees that grow in the rocky slopes. So quite interesting. We can examine there by using, I'm not going to enter into the details because of time, but we can explore the correlates between metrics like the multi-layer eigenvector centrality of each individual tree, explore what are the correlates with fitness variation and with seed dispersal success, etc. So those are aspects that we need to use to capture the whole biodiversity of interactions and try to explore those multi-layer networks to have a tunnel view, not to focus in a particular interaction, but to have the combination of interactions that, for which the local plant recruitment and local plant population persistence depends. And this is crucial and very urgent. As the IPBES report, we are facing not just and this is the message, central message of my talk, we are losing biodiversity, of course, but biodiversity is more than a species. It is also the biodiversity of ecological interactions. And we have those serious, somehow cryptic effects of losing those interactions, delays in forest regeneration, losses of carbon stock potential, losses of connectivity in metapopulation scenarios, or those examples of very rapid evolution in response to human-induced deforestation and overhunting. Let me finish by thanking my collaborators in the research group. I'm really privileged, and I thank their collaboration and support over the years that have been fundamental for the adequate development of all those projects. And also to my partners abroad, Cristina Garcia from the Holloway University, Royal Holloway University in London, Jordi Vasconte, University of Zurich, Jens Olesen, University of Aarhus, and Paulo Guimaraes and Mauro Galetti, University of Sao Paulo, in Sao Paulo City and Rio Claro, respectively, and Professor John Thompson in University of California, Santa Cruz. Thank you very much to all of you for your attention, and my appreciation also to the organizing committee for his splendid work in the organization and for privileging me with this opportunity. That, as John said in his previous talk, I don't forget. Thank you very much.
Thank you so much, Pedro. We have a few minutes for questions. Oh, yes, 1 minute? One question. We have time for one question.
Well, I'll be around here today and tomorrow, so I'm very happy to take any questions if you have. Thank you.
Actually, maybe I will ask a question, and then we'll stop. And please, afterwards, come and talk more to Pedro. There seems to be a general sense, particularly in the Canary Island data, that dispersing further is necessarily better. And a lot of the argument seems to revolve around that if we lose the tails of the distribution, we're in trouble. Now, clearly, seeds need to get away from the parent plant, but is there any real evidence that getting further and further and further is going to be better? What's the data suggest?
Yeah, I didn't like to imply that, but just because one reason, it is extremely important also to preserve the local populations, and the local regeneration by seed of the local populations depends on short-distance dispersal events. Those are fundamental for preserving the capacity of our population to persist in a given place. But then the long-distance dispersal events are extremely important, are going to be extremely important, for instance, in modulating the capacity of plants to respond to selective pressures involving extension of areas, colonization of new habitats in response to climate change. For instance, migration, altitudinal migration, or migration across different habitats will depend not on the local recruitment patterns that we can monitor in local populations, but on the long-distance dispersal events.
Thank you.